Okay, let's start our today's topic. As you know, we were discussing about nucleic acids. Yesterday, we had a brief introduction to nucleic acids. Now you know nucleic acids. They were discovered by Frederick Michel in 1871. He named them as nuclein. Then the name was changed to nucleic acid by Altman in 1889. Then it was Oscar Hertwig in 1884 who found the role of nucleic acids in transmission of hereditary characters. Right. Then in 1920, yesterday I told you 1950. In 1920, P.A. Levine worked out the chemical composition of these nucleic acids. Yesterday I told you P.A. Levine worked out chemical composition of nucleic acids in 1950. Actually, it is 1920. P.A. Levine in 1920 worked out the chemical composition of nucleic acids. He found that these nucleic acids, they are formed of nitrogenous bases, pentose sugars, and phosphoric acid, right? This helical structure of nucleic acids, it was first proposed by three scientists, Asbury, Wilkins, and Rosalind Franklin. Through their X-ray diffraction techniques, they found that these nucleic acids are in the helical form, right? But due to untimely death of Rosalind Franklin, the project remained incomplete. Then their work was carried ahead by Watson and Crick. And Watson and Crick in 1953, they proposed the double helical model of DNA, right? They worked out the structure of DNA. They found this DNA is a double helical structure, and for which they got a Nobel Prize in 1962. Now, today we are going to discuss about the structure of DNA as proposed by Watson and Crick. So, we are going to discuss about Watson and Crick model of DNA. Model of DNA. What is the full form of DNA? Deoxyribonucleic acid. So Watson and Crick proposed this model in 1953. So according to Watson and Crick, DNA is a double helical structure. It is formed of two strands. A strand is here known as a helix. So according to Watson and Crick, DNA is a double helical structure. It is formed of two strands. These strands, they are parallel to each other but in opposite direction. The two strands of DNA, they are parallel to each other but in opposite direction. That is known as they are in anti-parallel fashion. So according to Watson and Crick model of DNA, DNA is a double helical structure. It is a double helical structure. Double helical structure. That means a DNA molecule is formed of two helices. It is formed of two helices. Singular is helix, right? It is formed of two helices. These two helices of DNA, they are anti-parallel to each other. They are anti-parallel to each other. These two strands of DNA, they are anti-parallel to each other. That means they are parallel. This is one helix, and this is another helix. They are lying parallel to each other. What do you understand by parallel lines? No text, please. No text during the class. I will get the person removed from the group. Okay. There are two strands in a DNA. These two strands of the DNA, they are parallel to each other. What do you understand by parallel lines? Parallel lines are those lines, if projected forwards or backwards, they never meet each other, right? So these are the two helices of DNA. They are parallel to each other, but the direction is opposite. The direction of one strand, suppose, is 3 prime, 5 prime. Another strand is in opposite direction, 3 prime, 5 prime direction, right? So we say DNA is a double helical structure. It is formed of two strands. Each strand is known as a helix. These two helices, they are parallel to each other, but opposite in direction. That fashion is known as anti-parallel fashion. So they are in anti-parallel direction. They are in anti-parallel fashion, anti-parallel manner to each other, right? These two helices, they are joined to each other by the help of hydrogen bonds. They are attached to each other by the help of hydrogen bonds. These hydrogen bonds, they are formed between their nitrogenous bases. They are formed between their nitrogenous bases, right? So first, we will have a look at the physical structure of this DNA molecule, as proposed by Watson and Crick. So for that purpose, we need to draw a diagram. So we will draw a diagram first. Rub this much area. See how the diagram is drawn. So you have to draw a line like this first. You have to draw a line like this. Then you have to draw another line over it like this. Then you need to draw another line. Sorry. Let's start it like this. Then we need to draw another line over it. So this is a double helical DNA. It is a right-handed helix. So I will shade this to indicate that it is a right-handed helix. I am shading it to make you understand that this is a right-handed helix. That is why I am shading it. Otherwise, I don't recommend shading of diagrams in biology. Right? So these are the two helices. They are parallel to each other, but they are opposite in direction. Suppose this is the 3' end of one strand. This is its 5' end. This is the 3' end of this strand, and it is its 5' end. So they are opposite. They are parallel to each other, but opposite in direction. These two strands of DNA, they are the helices. Singular is helix. This is a DNA helix. This is another helix. This is another helix. These two helices, they coil around an imaginary axis. These two strands, they coil around an imaginary axis to assume this helical shape. So these two helices, these two strands, what are these two strands? They are actually sugar phosphate backbones. Each strand is a sugar phosphate backbone. This is one sugar phosphate backbone. This is another sugar phosphate backbone. So both of them are sugar phosphate backbones, right? That means they are formed of pentose sugar and phosphoric acid. These two sugar phosphate backbones, they are joined to each other by hydrogen bonds. between them. So these sugar phosphate backbones, they have attached to them nitrogenous bases. This is a nitrogenous base. This is a nitrogenous base attached to sugar phosphate backbone. They are attached to each other by the help of these nitrogenous bases. This is a nitrogenous base. This is a nitrogenous base. So hydrogen bonds are formed between the nitrogenous bases. These are the nitrogenous bases. Hydrogen bonds are formed between nitrogenous bases. So these are nitrogenous bases. And in between these nitrogenous bases, there are hydrogen bonds. There are hydrogen bonds. There are hydrogen bonds. How many nitrogenous bases are present per turn of the DNA? The distance between one shallow groove to another shallow groove. This is a shallow groove. This cut, which is not deep, this is known as a shallow groove. It is a shallow groove. This cut, which is not a deep, like this cut, which is small, it is known one shallow groove. This is another shallow groove. This is another shallow groove. The distance between two shallow grooves is known as one pitch. This length of DNA from one shallow groove to another shallow groove, or you can say from one deep groove to another deep groove. This distance is known as one pitch. This is known as one pitch. In this one pitch, DNA makes one complete turn. In this one pitch, DNA makes one complete turn. So if you are asked what is a pitch, you can say pitch is a one complete turn of DNA. Pitch is a one complete turn of DNA. And in the diagram, it can be represented by distance between two shallow. This groove is a shallow groove, this is a shallow groove. So distance between two shallow grooves, from one shallow groove to another shallow groove, it is known as one pitch. And this one pitch represents one complete turn of DNA, right? This is a deep groove. This cut is known as a deep groove. This cut is known as a deep groove. Similarly, here is another deep groove, here is another deep groove. This is also a deep groove. So there are deep and shallow grooves in this DNA, right? So distance from one shallow groove to another shallow groove, it is known as one pitch. And one pitch represents one complete turn of DNA. How many base pairs are present in one pitch? There are 10 base pairs in one pitch. 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. There are 10 base pairs per pitch in the DNA which is found in living organisms. The DNA which is found in living organisms, that is known as BDNA. That is known as BDNA. There are different types of DNA. There is one type of DNA known as AD, there is another known as B, another is known as cDNA, another is known as ddNA, another is known as zDNA. The DNA which is naturally found in living organisms, that is BDNA. In BDNA, there are 10 base pairs. 10 base pairs per pitch. There are 10 base pairs per pitch. What is a base pair? According to Erwin Shergoff, Shergoff's complementary base pairing rule. Complementary base pairing rule. Base pairing rule. The nitrogenous bases, adenine, it will always pair with the nitrogenous base thymine. You have learned about the nitrogenous bases. You know in DNA there are four nitrogenous bases: adenine, thymine, cytosine, and guanine. Adenine and guanine, they are purines. Cytosine and thymine, they are pyrimidines. You have learned about it, right? So according to Erwin-Chargaff's complementary base pairing rule, in this DNA, the nitrogenous base adenine will always pair with thymine, and cytosine, it will always pair with the nitrogenous base guanine. So this pairing of a purine base with a pyrimidine base, you know adenine, it is a purine base. Thymine is a pyrimidine base. Cytosine is a pyrimidine base and guanine is a purine base. So you will find it here in DNA that a purine base, this is purine, it will always pair with pyrimidine. This is the pyrimidine and it will always pair with the purine, guanine, right? So according to this Erwin-Chargaff's complementary base pairing rule, a purine base will always pair with pyrimidine and a pyrimidine base will always pair with purine. And the number of purines will always be equal to the number of pyrimidines. Or we can say the ratio between purines and pyrimidines is always equal to 1, right? So this is known as complementary base pairing rule of Erwin-Chargaff. He proposed this rule perhaps in 1947. I am not sure about dates. I have told you already, I forget the dates. So perhaps it was proposed by him in 1947, right? So according to complementary base pairing rule of Erwin-Chargaff, purines always pair with pyrimidines in DNA, right? So adenine is a purine base. It will always pair with a pyrimidine base, thymine. And cytosine is a pyrimidine base. It will always pair with a purine base, guanine, right? So this pairing between purines and pyrimidines, it is known as complementary base pairing rule, and it was proposed by Erwin-Chargaff in 1947. So according to this rule, the number of purines will always be equal to the number of pyrimidines. Or we will say the ratio between purines and pyrimidines will be always equal to 1. So how many such base pairs are present in a pitch? In a single pitch, in bDNA, there are such 10 base pairs. There are such 10 base pairs. Suppose it is adenine, it will be pairing with thymine. It is guanine, it will pair with cytosine. It is cytosine here, it will pair with guanine, right? It is thymine, it will pair with adenine, right? Similarly, it is thymine, it will pair with adenine. Cytosine, it will pair with guanine. Guanine, it will pair with cytosine. Thymine will pair with adenine, and adenine will pair with thymine, right? So in this way, nitrogenous bases, they are projecting on the two sides of these helices. This is the sugar phosphate backbone. Attached to this sugar phosphate backbone on this side is a nitrogenous base. This is also a sugar phosphate backbone. Attached to this sugar phosphate backbone on this side is a nitrogenous base. And hydrogen bonds are formed between these nitrogenous bases. Two hydrogen bonds are formed between adenine and thymine. Two hydrogen bonds are formed between adenine and thymine. And three hydrogen bonds are formed between cytosine and guanine. It is not double bond and triple bond. Some persons, they call them as double and triple bonds. That is incorrect. Between adenine and thymine, there are two hydrogen bonds. Between cytosine and guanine, there are three hydrogen bonds. So these hydrogen bonds between the projecting nitrogenous bases, they hold the two strands together. They keep the two strands joined together. And these two strands, they coil around an imaginary axis. So this DNA becomes a helical DNA. It assumes the shape of a helix. It assumes the shape of a coil, right? So this DNA, it has got a diameter of, this DNA has got a diameter of, it has got a diameter of 2 nanometers. This DNA has got a diameter of 2 nanometers. The length of one pitch from one shallow groove to another shallow, the length is 3.4 nanometers. What is the length of a pitch? What is a pitch now? The distance between two shallow grooves, the distance between two adjacent shallow grooves is known as one pitch. How many base pairs are present in one pitch? 10 base pairs. What is the length of one pitch of DNA? It is 3.4 nanometers. Since there are 10 base pairs per pitch, then what will be the distance between two adjacent base pairs? If it is adenine and thymine, cytosine and guanine, what will be the distance between these two adjacent base pairs? What will be the distance between two adjacent base pairs? What will be the distance between cytosine, this base pair and this base pair? What is the length of each? 3.4 nanometers. How many base pairs are present in per pitch? 10 base pairs. So 3.4 divided by 10. So the length, so the distance between two adjacent base pairs will be 0.34 nanometers. Right? The distance between two base pairs will be 0.34 nanometers. So this is the physical structure of DNA. This is the physical structure of DNA. Now you know this DNA, physically it appears as a double-stranded molecule. It appears as a double-stranded molecule. These two strands, they are anti-parallel to each other. If we project them forwards, they will never meet each other. If we project them backwards, these two strands are not going to meet each other. So they are parallel. They are parallel. But their direction is opposite. The direction of one strand is 3 prime, 5 prime. The direction of another strand is 3 prime, 5 prime. Right? So they are opposite in direction. We call that as anti-parallel fashion. We call that as anti-parallel fashion. So this is 3 prime. I think this is not visible in your diagram. So this is 3 prime end. And this is the 5 prime end. Right? So they are parallel to each other but opposite in direction. So what are these two strands basically? These two strands, they are basically sugar phosphate backbones. These two strands are basically sugar phosphate backbones. You know pentose sugar and phosphoric acid. They combine and they form the sugar phosphate backbone. Then attached to these sugar phosphate backbones are nitrogenous bases or nitrogenous bases. These nitrogenous bases, they are attached onto this sugar phosphate backbone. This is a nitrogenous base, this is a nitrogenous base, right? These nitrogenous bases, according to Erwin Chargaff's complementary base pairing rule, a purine base pairs with a pyrimidine base and a pyrimidine base pairs with a purine base. This is a purine base, adenine, it will always pair with pyrimidine thymidine. This is a pyrimidine base cytosine, it will always pair with the purine base guanine. So these are nitrogenous bases, and it is between these nitrogenous bases that hydrogen bonds are formed. There are 2 hydrogen bonds between adenine and thymine, and there are 3 hydrogen bonds between cytosine and guanine. So these hydrogen bonds, they keep the 2 sugar phosphate backbones attached to each other, right? So this makes the double helical model of DNA. Then we see here in the diagram, in this DNA there are some cuts, there are some grooves. This is known as a shallow groove. This is known as a shallow groove. Your diagram is totally incorrect if you are not able to draw this shallow groove and deep groove alternately. When you make the diagram of DNA, you fail to draw these shallow grooves and deep grooves alternately. One shallow groove should alternate with a deep groove. Then it should alternate with a shallow groove. Then again a deep groove, right? If you are able to draw it like this, your diagram is correct. So this narrow cut, it is known as a shallow groove. This deep cut is known as a deep groove. The distance between one shallow groove to another shallow groove is known as one pitch. It is known as one pitch. And one pitch marks the one complete turn of DNA. So from here, the DNA rotates like this. So from this point to this point, DNA makes one complete turn. And how many base pairs are present per pitch? As I told you, there are different types of DNA. The DNA which is naturally occurring in the cells of living organisms, that is bDNA. In bDNA, there are 10 base pairs per pitch, right? So there are 10 base pairs, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. 10 base pairs are present per pitch. What is the length of one pitch? It is 3.4 nanometers, right? Since there are 10 base pairs per pitch, then what should be the length between? What should be the distance between 2 adjacent base pairs? The distance between 2 adjacent base pairs will be 0.34 nanometers, right? So this is the physical structure of DNA. This is the physical structure of DNA. Now, to make you understand what is 3 prime and 5 prime, to make you understand what is the 3' end and 5' end, we need to have a look into its chemical structure. We will see how sugar phosphate backbones are formed and how these hydrogen bonds are formed between nitrogenous bases. So for that purpose, we need to draw the chemical structure of DNA, right? Should I rub this diagram? Is this clear so far to you? यहाँ तक समझ आया ठीक से? Good. Should I rub this diagram? Okay. Now we will have a look at the chemical structure of this. This was the physical structure. Let's make it here. I will try to make it here. Clearly visible to you. Where from the board is visible. From this. So I will start it from here. So this is a pentose sugar. Which pentose sugar is found in DNA? Which pentose sugar is found in DNA? Okay? It is deoxyribose. It is deoxyribose. So this is the pentose sugar. Is it visible to you? This is deoxyribose. Why it is deoxyribose? Because it lacks an oxygen atom at carbon second position. This is carbon first position. This is carbon second. This is carbon third. This is carbon fourth. And this is carbon fifth. You can see at carbon second position, it lacks an oxygen atom. That is why it is deoxyribose. It joins with a nitrogenous base. It joins with a nitrogenous base. Let it be adenine. Let me join it with adenine. I will try to join it with adenine. This is adenine base, right? This is adenine base. So how do they join? They join by the elimination of a water molecule at carbon first position. So this water molecule will be eliminated. This water molecule will be eliminated here. So this water molecule, it is eliminated here, right? And nitrogenous base and pentose sugar join, right? So when a nitrogenous base joins with a pentose sugar, what is formed? When a nitrogenous base joins with a pentose sugar, what is formed? A nucleoside is formed. A nucleoside is formed. So this nucleoside will be known as adenosine, and adenosine nucleoside will be formed. Now this adenosine nucleoside, it joins with a phosphoric acid molecule. It joins with a phosphoric acid molecule. Right? It is OH here. By the elimination of one more water molecule. So one more water molecule is eliminated. And this becomes now adenosine monophosphate. So it becomes one nucleotide. It becomes one nucleotide. Now this nucleotide, it joins with another nucleotide. It joins with another nucleotide. So I will remove this H here. By elimination of another water molecule, one more water molecule will be eliminated here. So it joins with another nucleotide. Let it be cytosine. Let it be cytosine. Okay. H, H, double bond. This is 4. This cytosine, sorry, it is double bond over here. It is double bond over here. So, cytosine, when it joins with, when this cytosine joins with the pentose sugar, it forms the nucleoside cytidine. Then it joins with another phosphoric acid molecule. It joins with another phosphoric acid molecule. Like this. It joins with another phosphoric acid molecule. Right? So when it joins with another phosphoric acid molecule, it forms cytidine nucleotide, cytidine monophosphate. So one nucleotide joins with another nucleotide. One nucleotide joins with another nucleotide. Similarly, many more nucleotides join, right? So it becomes a polynucleotide chain. It becomes a one polynucleotide chain. In this polynucleotide chain, what we have? We have got nitrogenous bases, we have got pentose sugars, we have got phosphoric acid. So that pentose sugar plus phosphoric acid, it forms the sugar phosphate backbone. It forms the sugar phosphate backbone. Pentose sugar plus phosphoric acid, it forms sugar phosphate backbone, right? Pentose sugar plus phosphoric acid. Pentose sugar plus phosphoric acid. It forms a sugar phosphate backbone. And attached to this sugar phosphate backbone are the nitrogenous bases. There are the nitrogenous bases, right? So this becomes a one polynucleotide chain. This becomes a one single helix of the DNA. So this is one helix. What is the direction of this single strand, this single helix of the DNA? I want to make you understand what is 3' end and what is 5'. This is the first nucleotide, it joins with the second nucleotide, then it will join with the remaining nucleotides, right? So here in the pentose sugar, this is carbon first, carbon second, carbon third, carbon 4, carbon 5. Similarly, here in pentose sugar, carbon first, carbon second, carbon third, carbon 4, carbon 5. You can see bonds are formed between phosphoric acid at carbon 3 and this CH2OH group at carbon 5, right? So this strand, which has got carbon 3 free, this phosphoric acid, it has got carbon 3, where the bond is not being formed. So this is known as 3' end. The end of the strand, where the last sugar is present, it has got carbon 3 free. So this is its 3' end. And the same strand, at the top, it has got carbon 5 free, where it is not joined with another nucleotide. So this becomes its 5'. Are you understanding what I am telling you? I am telling you that each of these strands has got two ends, a 3' end and a 5' end. So what is a 3' end? 3' end is that end of the strand where the pentose sugar has got this carbon third position free, where the bond is not being formed, right? This is the pentose sugar, it is not joined with any other nucleotide. So here the carbon 3 is free. So this pentose sugar where the carbon 3 is free, it is not joined to another nucleotide. This is 3' position. And in the same chain, at the top, where carbon 5 is free, which is not joined to another nucleotide, right? Here you can see carbon 5 is joined to another nucleotide, carbon 3 is joined to another nucleotide. But at the end, carbon 5 will be free, it will not be joined to another nucleotide. So this becomes the 5'. So the end which has got carbon 3 free, that is the 3' end, and the end which has got carbon 5 free, that is the 5' end, right? So in this way, many nucleotides, one nucleotide, another nucleotide, they join and they form a polynucleotide chain. So how many polynucleotide chains are present in DNA? How many polynucleotide chains are present in DNA? Come on, tell me. How many polynucleotide chains are present in DNA? I know I am taking your extra time. How many polynucleotide chains are present in DNA? There are two polynucleotide chains. This chain and this chain. These are two polynucleotide chains. But these two polynucleotide chains, they are opposite in direction. So I will tell you about it tomorrow, inshallah, since we are running out of time. So this is section for today. Try to study this DNA at your home from the book. You will definitely get confused. Wherever you will get confused, you will ask me tomorrow in the next lecture. Okay, thank you.